Hi everyone, welcome to the EOS imaging modality presentation. So for this, I'll be going through both the hip and lower limb analysis and give you a bit of an overview on this machine. So what is EOS? EOS was developed in France and utilizes X-rays at a much lower dose compared to conventional alternatives. It also results in full standing wide planar images, as you can see to the right, and utilizes a 3D construction software called STAIR EOS, which I'll be talking about in these slides. So you can see STAIR EOS to the right. We do have a computer within the bone imaging lab that allows you to do this analysis. And it basically allows you to do modeling workflows, mainly for the lower limb alignment, which you can see to the right here, and for pelvic parameters. So to do this, you would basically click the EOS 3D button here, which would prompt this window. You would then select whether you wanted to do the pelvic parameters or lower limb analysis, which would prompt this window, where you then select whether you start with the left or the right limb. Notice that whatever you select this first round, you'll need to go back and essentially repeat this process for the other. And then we can select either full 3D mode or lower limb alignment. Lower limb alignment is a more straightforward analysis, so it's going to be a much more condensed analysis compared to the full 3D model. So it starts us off with pelvic parameters, if that's what we selected. The nice thing is throughout this whole analysis, there'll be a little bar to the left, which will designate what steps you start with and what you're actually looking for within this stage. So for example, if we see 1, 2, 3 here, we know we're going to start the analysis in our lateral view, which will be with the first portion, the sacral plate, and then analyzing the femur head components here. Now with this one, it's important to note that you can get a little confused with the left and right orientation. For example, here, we'll know that the left side is here and the right side is here. But when we're looking at a lateral, it's much more difficult to designate which should be the left and right. You'll notice here that this is actually a mistake. In fact, this red left femur is actually presented here. A quick way to check whether you should have your left leg here or your right leg here is to understand whether this is a right lateral or a left lateral. So in this case, without the designation here, it is a right lateral. So in that case, we know that the bone that will be slightly wider is going to be our left bone. So in this case, the red marker for the left should be along this bone, and then the right one should be for this one. Our additional steps for this stage include also identifying the acetabula, which is right here, and this will just landmark for the rest of our steps. So the next stage is overall identification of the lower limb. So this is a pretty simple stage as you're just landmarking the major components. You also don't have to be very accurate in this stage, as the next few precise identification will allow you to fine-tune every aspect that we do here. So you'll first start with centering the femoral head, going to the trochlea notch here, and then a third of the way up of the diaphysis, which will be here, also known as our femur. We'll then repeat this on the tibia for the frontal view, and then repeat it on the lateral view. Once again, it will showcase exactly what order you're supposed to do every stage in, as seen to the left. Precise identification of the lower limb will then go and fine-tune everything we've just selected in the previous steps. So when we started by selecting our femoral head, here for this first stage, we can actually increase or decrease the diameter to better fit it in both the frontal and the lateral view. We also have another step, including making sure that it's centered for the third view of the diaphysis and other aspects as well. Now to the left, you'll notice there's a lot of different parameters. Depending whether you selected the lower limb alignment or the full 3D lower limb analysis, it'll depend on the amount of parameters you'll be able to analyze. So this is just a few of them. For the sake of the presentation, I won't be going through all of them. Another thing to note here is for the actual precise identification of the condyles. So you'll notice here that this is actually a really important stage for the overall analysis, as this is a really a big site of geometry for the knee. So there's a lot of different parameters here that we want to make sure we get a close fit to as well. You'll notice in the lateral view here, it might be difficult to distinguish which one is the right and which one is the left. But the important thing to notice here is that the software has a built-in tool that'll essentially landmark which is the left and right. And then essentially you have to go in and select that as well. So while it's a little bit difficult, if you actually mess these up, you will get a prompt that tells you that the condyles have been improperly reversed. So that's a good check there. Okay, and then this is the result display. So you can see to the left here that will have essentially all the parameters that we've identified. So this is after repeating the analysis for both legs, which is important to note here. Now this might be difficult to see, but you'll be able to tell in the next few stages how we can export this to both Excel and a more individualistic report. We can also see to the right here that we can get the 3D analysis. So this is essentially what the whole program does. It overlays a 3D model of the limb and then can show you how that would look in a visual space. So you can also export these views as well. This was after just segmenting the left leg, but you can get it for both as well. Okay, now if we want to export this data, we first have to make sure to save, which is this button here, and then we'll be prompted to select this table button. So if we select that, this will pop up. So this is a window that essentially summarizes all of the parameters that we've collected so far for both the right limb and the left limb. You can also click the lower limb button here, which will allow you to select more parameters or less parameters if you're wanting to streamline it. And then when you're ready to export to Excel, you can click this button. Now there is a folder on EOS that summarizes all of the Excel documents. However, if you are doing more fine-tuning analysis for your specific project or thesis, it might be worthwhile to create your own folder separate from this. That way you can collect all the files in one space. Another thing we can do is create a report. So to generate a report, we again want to make sure we've saved the model and then click this Add Report button here. We'll then click the Save Report button here as well. And once again, there is a folder on the computer designated for the stereo EOS reports. When you open the stereo EOS reports, this is one portion from the Word document, but essentially what it does is it tells you what parameters you've collected, what the value is for that specific analysis, and then a visual representation of what that parameter is alluding to, which can be really useful when you're learning these parameters for the first time. For length and knee parameters, they're also displayed here, so it'll have femur and tibia lengths and specific knee parameters like valgus and various angles and different mechanical angles as well. The last section is the torsions for the femoral, tibial, and femoral tibia rotations, so these can also be useful in your analysis. That brings us to the end of the presentation. If you have any questions, please contact Isabella to help you with your analysis.